So today we have a guest from University Hospital Aachen, Professor Liedtke, and he's the head of the research group in University Hospital Aachen. Is a strong focus on cell cycle, apoptosis, fibrosis, and HCC. So Professor Liedtke obtained his PhD in 1998 in Braunschweig Technical University. And after a postdoc period in Boston, he moved to Hanover, where he worked in the department of Professor Manz. And first he was a postdoc, after he was associate professor. And in 2005, correct, he moved to Aachen, University Clinic Aachen, and started to work in the department of Professor Trautwein. Professor Liedtke obtained his professorship in 2014, and he is the author of 75 publications and also a principal investigator in the German Collaborative Research Center Fibrosis and the Gut-Liver Axis. And I have two more words to say. So Professor Liedtke, I know him for 16 years now. So I started first as a young trainee, then a PhD, postdoc, and a co-PI in his lab. And I can say like many words about support and help and kindness and knowledge, no? But I just want to say I always name him my Dr. Vater. So Dr. Vater is, if translated from very old German, is Dr. Father. And this is what he is. This is what is he for me. So please, Professor Liedtke.
Good afternoon, everybody. And thank you, Julia, for this kind and nice introduction. Hello, Barcelona. Yeah, it's really a pleasure to be here in Madrid, and I really enjoyed all these talks and seminars that we had during the day, and it was really nice to see all these projects and this enthusiastic data that has been presented, and I thank you very much that you shared your data with me. So, before I start with science, I just want to show you where I come from. This is a picture of the University Hospital in Aachen, which is now a listed building, a national monument, because this kind of architecture this reminds us to industry and yeah, it's unique. It has been established in '85, and the only building that is similar is the Centre Pompidou in Paris, and these two buildings represent its own style of architecture. It's special. And what also is protected is the green color inside. This is not a subway station. This is just our main entrance hall. And the good thing in this building is that everything, including patients and diagnostic therapy and basic research, is all located under one roof. This is just my introduction. And now I want to introduce what I will talk about today. This is my agenda. And first of all, I will give you a very short overview about the cell cycle machinery, which is known in the textbook. And I will also show you that this is only half of the truth. Then I will give you a small overview about the lessons that we gained from knockout studies about cell cycle regulation in mammals, and especially in the liver. And I will also tell you the story that we had discovered when we tried to figure out the core machinery of the cell cycle in the liver, which was a very hard time. And our first positive results for a unique function of one cyclin and one liver cell. I will try to convince you that cyclin E1 is a druggable target in liver fibrosis. And I will also show you some unexpected data in the course of HCC development. And finally, I would like to discuss with you if it would make sense to interfere with the cell cycle regulators in some therapeutic approaches. Okay, so maybe this is just one introductory slide. You all are familiar with pathogenesis in liver, and we all know that the liver is a detoxifying organ, and which is exposed to a lot of stimuli such as virus infections. We have the phenomenon of the poor nutrition, and we also have the problem with the exposition to drugs and toxins. And I want to focus on the role of proliferating cells in liver homeostasis and in disease. And during homeostasis, we know that the liver is working very hard, is trying to detoxifying things. And of course, hepatocytes are dying. Usually this is not a problem because the remaining hepatocytes have the strong regeneration capacity and due to the cell cycle activity in hepatocytes. The next step, if we have a constant stimulation with toxins or inflammatory mechanisms, some other cells come also into the game, which have to proliferate in order to respond to the NOx cells. These are, for instance, the hepatic stellate cells, again hepatocytes, and maybe several other cell types. And finally, I will show you some examples of proliferating cells in the course of hepatocarcinogenesis. Again, this is just an introduction. And so I will focus on the one core complex in cell cycle regulation, which is a cyclin CDK complex, because we believe that the cell cycle, not only in the liver, but in general, is basically controlled by cyclin-dependent kinases and the regulatory subunits, which we call cyclins. And we have in mammals, we have many of them. And I will give you this textbook information, what we believed some years ago, how they contribute to the cell cycle regulation in the liver, but also in other cells. We start with the resting hepatocytes. We call the state G0 because they are not really active in the cell cycle. And upon some mitogenes, which can be secreted by Kupffer cells, for instance, this can be mitogenes, growth factors, STAT3 activation, this kind of things. Cyclins from the D-type are activated by transcriptional mechanisms. And these D-type cyclins can interact with a certain group of CDKs, which are called CDK4/6. And this initiates a subset of events that trigger the release from G0 into the active phase of the cell cycle. And then in the next step, the replication of the DNA is initiated, and we believe that cyclins of the E-type are responsible for this mechanism together with the kinase subunit CDK2. This is what you find in the textbook. And the DNA synthesis itself is then regulated by a combination of CDK2 with cyclin A. In other words, CDK2 can change its partner. And then finally, the mitosis and the cell division, the cytokinesis, is basically controlled by B-type cyclins together with another CDK, which is CDK1. So this is what you find in the textbook, and this is not really true. Because in the last decade or so, or maybe the last 15 years, many people and groups have shown that the genetic knockout of most of these cyclins and CDKs has mostly no effect. This was at the beginning very frustrating. This is an overview. If you delete any of the D cyclins, you get at best minor effects or minor defects. The same is true for CDK4, CDK6, CDK2, which was a big surprise because some time ago people believed if you delete these kinases, this could be a very promising approach to treat, for instance, cancer. But this is not true. The same is true for E-type cyclins. So when Julia was starting in my lab, she deleted or she analyzed mice with a deletion of cyclin E1 or E2. And I will show you that they have at best moderate effects. So the only exemptions are cyclin A2 and the B-type cyclins and CDK2. If you delete them in the embryonic stem cells, then the mice are not viable. And there has been a strong discussion why this is the case. Why are most of the components dispensable and some not? And there was a nice review from the group of Mariano Barbasit here from Madrid, and they proposed that maybe this has something to do with the evolution of the cell cycle from yeast to man. And in yeast, the situation is very simple. We have one CDK, which is named CDC28 and which shares strong homology to CDK1. And in yeast, you only need one CDK and many cyclins, and they regulate the complete cell cycle. And so the Barbasit group postulated that maybe the same is true in mammals and in men. So we have one core CDK that is responsible for the major cell cycle. And in some cases, can I use? No, it does not work. In some cases where you have specialized cells, maybe you need you need other cyclines and CDKs to regulate specific functions. But I will show you that this is only true in some of the cases. So now I will switch a little bit and come to some knockout studies. So by the way, what is the goal of modulating cell cycle components? I will give you some overviews, what we expect and what could be beneficial and what could be maybe harmful. So if we target the cell cycle in general tissue cells, yeah, one would expect, and this was what people were expecting when they started these knockout strategies, one would expect developmental defect. Oh, this is now hidden by the by the Barcelona image. Yeah? Okay, of course we do not want to have developmental effects if we interfere with the cell cycle. We want to perform a therapy. So this has to be taken into account. Yeah? Regarding hepatocytes, and if we interfere with the cell cycle, one would expect impaired liver regeneration. Yeah, if we perform a therapy, this is also not what we wanted, but we have to consider that this could be a negative side effect if we interfere with the cell cycle. So what about hepatic stellate cells? They are main cells for liver fibrogenesis, and if we interfere here with the cell cycle, one would expect or hope that we could protect our subject from fibrosis development, because less activated stellate cells would mean less fibrosis. And for immune cells, it's a little bit more complex, but one would expect that if you could interfere with the cell cycle in some immune cell subpopulations, maybe you have a beneficial effect on the inflammatory response. By the way, the regulation of cell cycle in immune cells is poorly understood. Of course, we know some cell type populations where proliferation is very important, for instance, T cells and B cells, that we need it. But for Kupffer cells, for example, it's still under debate if they really proliferate a lot or if they just develop from progenitor cells. And finally, we have hepatoma cells. And here, of course, we have a very strong interest in regulating modulating the cell cycle regulation, because this could really help to treat liver cancer. Okay, and I will go through these different models, and then at the end we can discuss if this really makes sense to interfere with the cell cycle regulation in the liver, or if this maybe not sufficient. Okay, and I will start a little bit in the early years of our lab, when also Julia came into the lab. We started with some analysis in the partial hepatectomy model, and we discussed this already today. I think that you all are familiar with hepatectomy. And we started with conditional mice that had a knockout for cyclin E1. And we had the idea, if we knock out cyclin E1, yeah, we can block the complete cell cycle in the liver. And this was not true. I here have the scheme where I indicate that cyclin E1 is absent, and all the other components of the proposed cyclin CDK complex are still shown on the slide. And in parallel, there's another E-type cyclin, which is named cyclin E2. And we believed that this also should have some effects. But you can see here that single knockout of either cyclin E1 or E2 had at best a very moderate effect. Yeah, we found a 4-hour delay in the onset of DNA synthesis in the liver, which is really minor. But at the end, the animals all survived and all had normal restored liver mass after the end of the experiment. And the second surprise was when we looked at cyclin E2 knockout mice, they even showed hepatomegaly, which was unexpected. And I will not explain in detail why they have hepatomegaly, but otherwise the livers were normal. Yeah, so this was the first. We were quite disappointed. We published this data in Gastroenterology, but we expected a much stronger effect. Then we switched to CDK2, which is a proposed binding partner of E-type cyclins, and performed a hepatocyte-specific CDK2 knockout. We got this mice from Mariano Barbacicchio from Madrid. And you can see here that one would expect that cyclin E1 and E2 are still expressed. CDK2 is absent, so the kinase binding partner is missing. And this was even more striking because there was absolutely no effect. Yeah, you can see here that the kinetics of DNA synthesis is absolutely identical to the one of control animals. And if you look at the liver mass restoration, there was no difference. So we can keep in mind that CDK2 is fully dispensable for liver regeneration in mice. Yeah, and this asks the question whether CDK inhibitors, pharmacological inhibitors which are available, yeah, are suitable to interfere with the liver homeostasis or not. And to complement these findings, I want to shortly mention some data of other colleagues. So the group of Philip Caldes analyzed CDK1 knockout mice, because I showed you this hypothesis from the Barbasit group, which proposed that CDK1 can maybe take over all the functions of the other CDKs. And they analyzed CDK1 knockout mice, and they found that they can also regenerate. Yeah, they were even able to perform normal DNA synthesis, but they had a defect in cell division. But the liver regeneration took place without any problems. There you can see here that the hepatocytes are somewhat smaller. And then, in addition to this, the group of Mariano Barbasit here in Madrid, they performed double knockouts for CDK4 and CDK2. And they also find that these mice are completely happy. Yeah, so even with a knockout of 2 CDKs, yeah, the cell cycle in the liver can occur, and you see normal liver regeneration. Okay, this might be frustrating, but still, we did not give up. So we started to dissect the cyclin E CDK2 complex in liver regeneration step by step and performed double and triple knockout strains. So this is an example where we deleted cyclin E1 and CDK2. And here you can see the first effects. Yeah, so on the left-hand side, you see that the DNA synthesis, shown here by BRDU staining, yeah, is dramatically reduced, and also the liver mass restoration is strongly affected. But I have to mention that still we do have some DNA synthesis in the liver, so it's not completely blocked. All the mice survived, and part of the liver mass restoration was done by hepatocyte hypertrophy. Yes, this we could also measure and quantify. Yeah, but there must be some safety mechanisms that really prevent that the mice die by incomplete liver regeneration. Yeah, then we took another approach. We deleted cyclin E1 and E2. We got very similar results. Yeah, maybe you can say that CDK2 can interact with some other cyclins and perform some residual mechanisms. Yeah, here again we have reduced DNA synthesis, impaired liver mass restoration, but the mice all survive. And finally, we generated triple knockout mice with depletion of cyclin E1, cyclin E2, and CDK2, and we got no additional effect. Yeah, so here in the diagram, you can see all the combinations that we tested, and we can summarize that in order to get an impaired liver regeneration, you need at least to delete cyclin E1 plus another member of this family. This can be either cyclin E2 or CDK2. Otherwise, you will get no effect. Okay, with this, I will summarize our data on the liver regeneration. We learned a lot. And we can state that the cell cycle machinery in liver regeneration is highly redundant and has a strong mechanistic overlap. So deletion or pharmacological inhibition of single key players has at best minor effects. Yeah, so this is summarized here. Single genetic knockout of any of these players has at best a minor role. Yeah, you need at least to delete a combination of 2 of these players. Cyclin E1 is maybe the most important guy, yeah, together with another partner of this complex. And even if you deplete the complete cyclin E CDK2 complex, yeah, you will get survival and liver regeneration of these animals. Okay, so this is a cartoon that I will show from time to time just to remind you where we are and what we already found. So in the first step regarding liver homeostasis, yeah, we have shown some important function of cyclin E and CDK2. And I will now switch to the liver fibrogenesis. So the first question is, why should liver fibrogenesis require the cell cycle machinery? Yeah, so when I started with liver fibrosis research as a cell cycle biologist, I was asking exactly this question. And then we did this simple experiment. We treated mice with CCL4 and then did a simple Ki67 staining. And what you can see here are the brown nuclei that are indicating proliferating cells. The first cell type we already know, these are the hepatocytes, and I have just shown you that they require E-type cyclins and CDK2 for regeneration, also as a response on CCL4 treatment. And the other guys here, also encircled in red, are hepatic stellate cells. And we believe that the resting hepatic stellate cells are quiescent, but if you stimulate them with profibrotic agents such as CCL4 or viral infection, whatever, we know they get activated and they start to proliferate. We don't know exactly if the activated cells proliferate or if it's quiescent cells that first proliferate and then get activated, but at least there is some cell cycle activity as shown here. So our idea was, if we can manipulate the cell cycle in hepatic stellate cells, maybe we can cure liver fibrogenesis. And so altogether, I think that liver fibrogenesis is very well associated with cell cycle activity. And this is proof done in wild-type mice after CCL4 treatment. And wherever you see these arrows, there are orange spots which are cyclin E expressing cells in the scars of the fibrotic tissue. And this motivated us to go further into detail. And we took our cyclin E mice and characterized them in fibrotic models. And this is another study which was basically performed by Yulia Nevzorova, showing that a constitutive knockout of cyclin E1 in all body cells protects from liver fibrogenesis, at least in the CCL4 model. So now I show you some unpublished data which are at best 6 or 8 weeks old, because the key question was still, is this what we hypothesized? Is this profibrotic function of cyclin E1 really related to hepatic stellate cells or to other cells? And maybe you know that it's not that long ago that HSC-specific CRE mice are on the market, made by Robert Schwabe, and he was so nice to provide us these mice. And we performed the following experiment. First, we generated conditional knockout mice that have only a cyclin E1 knockout in hepatic stellate cells. And then we took advantage of the so-called DN-CCL4 model, which first induces fibrosis with CCL4, and due to the DN injection, the mice also end up with HCC. And I'll show you only the first part of the data. This is after a couple of weeks, showing that deletion of cyclin E1 only in hepatic stellate cells give rise to dramatically reduced liver fibrosis in mice. And this is the first proof that really the stellate cells are the target cells of this cell cycle activity. Then the question is, of course, is this profibrotic activity of E-type cyclins dependent on the canonical interaction partner CDK2, which was not known at all. And therefore we performed a similar experiment. We generated CDK2 mice, which lack CDK2 only in hepatic stellate cells, and we get a similar result. So the fibrosis is largely reduced. In other words, we now believe that the cyclin E CDK2 kinase complex in hepatic stellate cells is very important for the onset of liver fibrosis. So now the question is, can this antifibrotic potential of cyclin E1, can this be targeted? Does it make sense, or is it possible to target cyclin E1 in a way that we prevent liver fibrosis? And in the next couple of minutes, I want to convince you that the answer is yes. In order to test this idea, because there's no pharmacological inhibitor available for cyclin E1, only for CDKs, we took advantage of siRNA. And at first we tried to optimize a protocol to deliver siRNA in the liver. So we took advantage of a fluorescently labeled siRNA that we injected in mice, and then did some kinetics in order to figure out where it goes. And this is a nice picture that ended up as a cover in Hepatology, showing that most of the siRNA ends up in the liver and remains stable in the liver for at least 1 week. This was very promising, but of course we also needed to know in which cells which cells can be targeted as siRNA. And this is the result, which is very promising. We could figure out that 90%, 95% of the HSCs can be targeted by siRNA if we deliver it through the tail vein. And regarding hepatocytes, we reach around 70%, and we also reach a large proportion of CD45 positive cells and also a lot of endothelial cells. So regarding the liver, it is maybe a systemic approach, but we can at least reach the important players. But does it work? In a first pilot experiment, we did only a short-term exposition. We already knew from our previous data that if we inject CCL4 to mice, we get a very strong cyclin E peak after 48 hours. So we took wild-type mice and 3 different cyclin E1 siRNAs. We injected CCL4 to the mice and analyzed the animals 48 hours after CCL4 and after siRNA treatment. And here shown in this one sample was highlighted with the red arrow, shows you that one of our siRNA molecule was very potent and could really reduce the cyclin E expression to almost baseline level. This was very promising. And on top of this, we could show that also the protein expression of cyclin E1 in these mice was drastically reduced, and even better, retinoblastoma protein, which is a target of cyclin E1 CDK2 kinase activity, is no longer phosphorylated after siRNA treatment. In other words, we can even target the substrate of the cyclin E CDK2 complex, at least in the short-term experiment. Then in the next step, of course, we wanted to make a step forward in the direction of fibrogenesis. And therefore, we took advantage of the following protocol, where we first injected a strong dose of cyclin 1 siRNA. Then and this was repeated every week, once a week. And then we injected the mice 3 times a week with CCL4 to induce medium-sized fibrosis. So this we call a prevention model. And some of the results are shown here. First of all, again, we can show that a delivery of siRNA once a week is sufficient to drop the cyclin E1 expression for at least 1 month to baseline levels. And again, you can see that on the protein level, cyclin E1 is also dropped. And again, we can see that one of the major targets is no longer phosphorylated. Regarding fibrosis, I hope that I convince you that on the very right-hand side, the fibrosis level of the siRNA-treated mice is really strongly and significantly reduced. And I can tell you, if you prolong this CCL4 treatment for up to 6 weeks, the differences are even stronger. Yeah, so basically in a prophylactic model, this siRNA treatment is really antifibrotic. But this is not what the physician wants to do. The physician gets the patient, he makes the diagnosis and see, okay, my patient has fibrosis, and now I want at least to prevent the fibrosis progression. In other words, we first have to start with the fibrosis and then do a treatment, and then hopefully we can get a benefit out of it. And this is what we did in the next step. We first induced the liver fibrosis. Okay, I agree, it's a slight liver fibrosis. We injected CCL4 for 3 weeks, and then we started the treatment with siRNA once a week and continued with CCL4 for 3 additional weeks on top of that. And the result is shown here, and the quantification is shown on these diagrams, basically demonstrating that we at least can halt the fibrosis progression in this model. So the fibrosis level remains on the level that we had when we started the therapy. And we hope that we can even improve this outcome if we prolong this experiment one day. So now is again the question of the effector cells. I showed you that with our siRNA, we targeted a lot of cell populations, hepatocytes, stellate cells, endothelial cells, and we wanted to figure out which are the most important populations. And I cannot really answer this question precisely. This data I already showed you. Yeah, we have convincing data that the HSCs are very important. So we definitely have to target HSCs. This is without question. So then we had a look at the CD11b myeloid cells. And in our siRNA experiments, we could observe that in the control mice, we had also a strong proliferation of CD11b positive cells, which were significantly reduced after siRNA treatment. So we cannot exclude that also proliferation of this cell population acts in a way or supports the fibrogenesis. We need further data to really prove this, but we are convinced that at least the targeting of this myeloid cell population is not harmful. And in addition, we also tested if hepatocyte-specific knockout of cyclin E1 could also contribute to this antifibrotic effect. And in fact, this is the case. When we treat hepatocyte-specific cyclin E1 knockout mice with CCL4, we also get a very slight, but we do get a protection. So we now believe that, or the other way around, when we submitted this manuscript, the reviewers criticized that this was a systemic effect and not specific to HSCs. But now we do believe that because it is not specific but targets many cell populations, this effect is so convincing. So the good thing is that with this siRNA we target only the liver and not the other organs. But I really believe that at least targeting of myeloid cells, HSCs and hepatocytes all contributes to this antifibrotic effect. And in the first chapter I showed you that deleting cyclin E1 in hepatocytes is not harmful at all. So therefore we believe that this could be a potential therapeutic approach in the future. And we hopefully have a nice discussion after the talk about the pros and cons. Okay, I want now to summarize this fibrosis part. I hope I could convince you that liver fibrosis is really associated with cell cycle activity, at least in HSCs, hepatocytes, and myeloid cells. We also found that cyclin E1 turned out to be essential for fibrosis initiation. And we have some data showing that an siRNA-directed approach could be suitable to really help in a therapeutic treatment. And we have some data showing that this systemic application that has been criticized before does not seem to mediate harmful effects, but is even beneficial in treating the liver fibrosis, at least in mice. Okay, so in other words, we have moved one step further in the direction of liver fibrosis, and I hope I could convince you that cyclin E1 and CDK2 play an important role in the onset of liver fibrosis, but also for the fibrosis progression. So, in the final step, I want to show you some data regarding the role of E-type cyclins and CDK2 for HCC development and later for HCC progression. And here we also started with a simple prevention model using constitutive knockout mice. And we discussed today, so you are all familiar with the DEN model. So we took 3 different mouse strains, deficient for either cyclin E1, cyclin E2, or CDK2. We treated these mice with DEN once, and then analyzed mice at the age of 24 weeks, where we have at best small, very small liver nodules with a diameter of 1 millimeter or small premalignant liver lesions. And we analyzed the mice at the age of 40 weeks, where they have solid HCC nodules. And so we refer to these phases as tumor initiation up to 24 weeks and tumor progression up to 40 weeks. Okay, and these are the key results. You can see that the DEN works. In wild type, we get strong tumorigenesis. The same is true for cyclin E2 knockout mice, which was very surprising. But on the other hand, if you look at the cyclin E1 mice, they are not only significantly reduced, it's almost black and white. So you barely find, altogether we found 9 livers that had some tumors, but most of the livers were tumor-free. So it was very convincing. And then we came up with the question, is this maybe model-specific? Is this only true for the DEN model? And to exclude this, we crossed our cyclin E1 knockout allele into MYC mice that have been presented today in the seminars also. And we can show a similar effect. So double transgenic mice lacking cyclin E1 and expressing MYC are also protected from hepatocarcinogenesis. Then the question arises, why do at least some of the cyclin E1 deficient tumors grow? This was also a question of a reviewer, and we had a hard time to answer this, but maybe I can contribute something to this important question. So if you look at wild-type mice that have been treated with DEN, we found a very strong induction of the cyclin E1 gene expression, but we did not observe induction of cyclin E2 gene expression or cyclin A2 expression. Maybe you remember cyclin A can also interact with CDK2 and maybe compensate. They are both quite silent, and if we look at the right-hand diagram for the cyclin E1 mice, you can also see that neither cyclin E2 nor A are significantly induced. But when we look in more detail and really focus on the mice, on the cyclin E1 knockout mice that had some tiny residual tumors, we found some cyclin E2 expression in this encircled area, here shown in brown, yeah, which indicates that there is at least a little bit reactivation of cyclin E2. And we tried to prove this in more detail, so we isolated cytoplasmic and nuclear extracts, and we could really detect in cyclin E1 deficient livers some residual re-expression of cyclin E2, here shown as a red arrow, in this tissue. And therefore we conclude that the HSC initiation in cyclin E1 knockout livers can only occur if we have this untypical re-expression of cyclin E2, yeah, which usually is not found in our tumor samples. So HSC initiation is not related to cyclin E2 induction in standard samples. So again, we wanted to answer the question, is this dependent on the interaction partner CDK2 or not? Because usually the textbook says cyclins and CDKs form a complex and then act as a kinase. To this end, we had a look at hepatocyte-specific CDK2 mice, and as you can see here, we got very similar results. If you deplete CDK2 in hepatocytes, the tumor initiation and the number of tumors is dramatically reduced. And this is true, although the gene expression of cyclin E1 is still there. You can see this on the blue bar diagram. Yeah, we do have cyclin E1 expression. The binding partner CDK2 is missing, and you have no HCCs. This indicates that this effect is really dependent on the complex formation of cyclin E1 and CDK2. If one of the partners is missing, the HCC initiation does not take place. Yeah. Okay, and now I come to another part of the story, because we want to better understand the underlying mechanisms. Because we still had no clue what are the cyclins and CDKs doing. And to this end, we did a novel approach. For us, it was a novel approach, because we tried to isolate the hepatoma cells. To this end, we used floxed cyclin E1 mice. We initiated HCCs by treating them with DEN. And then after 40 weeks, we isolated solid HCC nodules, and we also isolated surrounding non-tumorous tissue, and tried to plate the cells and cultivate them in order to immortalize them, and this worked. Here is shown that with these animals and cells, we still have a wild-type cyclin E1 available. Yeah, and then we plated the cells, and in order to delete cyclin E1, we delivered an adenovirus expressing Cre recombinase in combination with GFP to track these cells. And then we performed a cell cycle analysis of the cells in order to figure out what's going on and what is the mechanism of tumor prevention. And yeah, these cells, cyclin E1 is then deleted. And what came out was that we get basically two populations of cells that behave completely different. The cells from the precancerous tumor-free tissue, I will refer to them as pre-CL cells. They were slow growing, and the cells from the large HCC nodules, they were fast growing and rather aggressive. And this is a typical growth curve. The gray curve is from the pre-CL cells, and the black curve is from the aggressive HCC cells. And they also have a very different expression profile. The pre-CL cells have low expression of AFP. The HCC cells are high in AFP. Yeah, we have a distinction between moderate versus high cyclin E1 induction, and the same is true for cyclin E2 and for cyclin A2. So we can summarize that the tumor-derived cells are more aggressive. They have induction of many cyclins and pro-tumorous markers, and the residual cells from the surrounding are more slow-growing. Okay, so what happens if we delete cyclin E1 in these cells? Again, we had two outcomes. In the pre-CL cells, we found signs of apoptosis. And this is shown here in the FACS diagram by this strongly increased subpopulation of sub-G1 cells, which indicates reduced DNA content. You can also see in the growth curve that after 2 days, the cell population collapses. So there's no doubling of the cells, but the cell number decreases after 2 days. The outcome for the HCC-derived cells is completely different. Here we have at best a small delay in cell growth and almost no apoptosis. So is this really apoptosis? What happened to the cells? And here I want to show you some live imaging. On the left-hand side, I show you the cyclin E1 flox cells that just received a Cre-expressing adenovirus, nothing else. And please look at the areas with the red arrows. You will see some proliferating cells. They have a normal cell cycle. On the right-hand side, you should try to figure out the differences. I try now the movie, and it should be very obvious. On the left-hand side, if you look carefully, some of the cells will duplicate or divide. And the right-hand side, the cells are collapsing. And this is a movie that we did for 36 hours. So we gave the adenovirus, then overnight we washed the supernatant away, and then we started with a movie for 36 hours. And after 36 hours, the right-hand population was almost gone, and the left-hand population was doubled. So this really indicates that acute deletion of cyclin E1 in this pre-malignant hepatoma cells really induces apoptosis and prevents tumor nodule formation. Okay. So now we wanted to understand in more detail what are the properties of these two populations, because this is also important for diagnosis and prediction. Once we have a signature, maybe we can predict which patient has a better outcome than the other one. So in order to figure out this, we did some standard measurements by qPCR, but we also did transcriptomic studies. So the first thing that came out was that the pre-CL cells have, I showed you this already, moderate cyclin E1 expression, and the HCC cells have strong expression. If we delete cyclin E1, of course, it's gone. But this, in HCC cells, this has no effect on the cyclin E2 expression, and also no effect on the cyclin A expression. So they don't care. So they have no cyclin E1, but they can grow with other cyclins and are cyclin E1 independent. And this is basically the pattern. The cyclin E dependent, I will call them so in the future, the cyclin E dependent cells, they have a moderate increase in cyclin E1 and in E2, and a moderate increase in cyclin A2. The cyclin E1 independent HCC-derived cells, they have a strong upregulation of E1, E2, and cyclin A2. This is the difference. And then we performed transcriptome analysis by next generation sequencing, and we found some surprising results. Of course, we expected that HCC-related genes are upregulated in these HCC cells, and cell cycle-regulated genes, this we already know. But we also found that several DNA damage response pathways were extremely upregulated in the cyclin E1 independent cells. And this was surprising, because one would expect the other way around. So they have an incredible strong activation of DNA repair machinery. And so from all this transcriptome data, we extracted a signature. This is only part of the signature of genes that are really typical for this signature, which are, for instance, ACT2, MSH2, RAD21, and E2F1. So this ACT2 is involved in DNA damage response and cytogenesis. MSH2 is a typical DNA mismatch repair gene. RAD21 is a double-strand break repair gene. And E2F1 is involved in general cell cycle control, but also in DNA damage response. So now I want to show you how this can be translated into the human situation, because then we went one step further and looked at the TCGA database and looked for related gene expression in patients that have already been characterized for the HCC signature. And you can see here that the patients that have a strong cyclin E1 expression have a very poor, significant poor outcome, which is also true for cyclin E1 and the combination of cyclin E1 and cyclin E2 upregulation. In other words, the cyclin E1 and E2 expression pattern is already indicative of the outcome for the patients. But now, maybe you remember the signature that I showed you with ACT2, MSH2, RAD21, and E2F1. This was really amazing that the patients that had high cyclin E2 expression and a poor outcome had already correlation with the high expression of ACT2, MSH2, RAD21, and E2F1. In other words, the signature that we found in our mouse cells for the most aggressive hepatoma cells can also be found in patients with a very poor outcome. So this means that the pattern in mice and men is at least similar. This is a big surprise, and now we should discuss if this signature can be used for future characterization of HCC patients. Okay, so now I want just to summarize this part of the finding. I wanted to show you that cyclin E2 is strongly overexpressed in a subpopulation of cyclin E1 independent hepatoma cells in mice. That in men, HCC patients with high cyclin E2 expression define a population with worse prognosis, and that this prognosis is also associated with the signature that we've already found in mice, with overexpression of ACT2, MSH2, RAD21, and E2F1. It has to be shown if this signature is somehow valuable for characterizing patients, and maybe if we want to treat one day or try one day a cyclin E1 therapy in a subgroup of patients. So I will make this very short. A few months after our study, two interesting papers came out which basically confirmed our findings and went into more detail, published in Nature Communications and also in Gastroenterology. This is interesting stuff to read, and if you have all this information, maybe we better understand what's going on. And I want now to ask the question, what about CDK2 in liver cancer progression? And this is also very surprising. We did the same strategy. We isolated cells from tumor nodules and from surrounding tissue of CDK2 deficient mice. We get similar patterns. We get a similar subgrouping of CDK2 dependent and independent cells. And we can sorry. And we can say that maybe CDK2 is important for the initiation of HCC, which is also shown in the mouse model. But if you have this cells from the isolated tumor nodules, it's dispensable. This was our hypothesis. And now we switch again to the patients. What can we say about CDK2 in patients? This was also very surprising. So we analyzed the CDK2 expression pattern in all groups of patients, in patients with high cyclin E1 and E2 expression, in patients with high E1 and low E2, and so on. And I show you this data, and basically it demonstrates that the patients that have either strong or low CDK2 expression have the same outcome. So CDK2 is not relevant for the outcome of HCC patients. The cyclin E expression is. So in other words, expression level of CDK2 does not affect the survival of HCC patients. And again, this is important if we are discussing that CDK2 inhibitors are on the market and could be used for therapy. This will not work. Okay, and again, we have the problem. This was all prophylactic what we did before. So we started with knockout mice and then induced HCC, but the physician wants to treat the patient with the HCC diagnosis. So in order to address this problem, we took advantage of MXCRE mice where we can induce the knockout of cyclin E or CDK2. So we followed this scheme. We had MXCRE cyclin E1 floxed mice or MXCRE CDK2 floxed mice. We injected DEN, and after 24 weeks, when we have the tumor initiation with the small liver nodules, we treated the mice with PIPC, and this gives rise to interferon induction and CRE expression and knockout of our target genes. And then, after 40 weeks, we sacrificed the mice and looked and quantified the HCC number of HCC nodules. And the data is shown here. On the left-hand side, I show you the data for the cyclin E1 knockout, and you can see here that there is a significant reduction of the number and size of tumor nodules if we delete cyclin E1 in the course of HCC development. And now the big surprise, when we do the same with the CDK2 knockout mice, there's no difference. There's no protection. We have the same number of tumor nodules. Yeah, it simply doesn't matter if CDK2 is there or not. So it's absolutely dispensable for HCC progression. This is experimental proof. Okay, and I now coming to the end and showing you part of our model, what we now believe what's going on in HCC cells. So if you stimulate the liver with pro-carcinogenic NOX, in the first step, which is CDK2 dependent and cyclin E1 dependent, yeah, you get a transformation into a precancerous stage of hepatocytes. And this is associated with a large number of double-strand breaks. I did not show this data, but we can prove this. And then we have two populations. The one population is the cyclin E1 dependent, or I call them cyclin E1 addicted hepatoma cells. They are characterized by a signature with low cyclin E2, low cyclin E1, overall decreased cell cycle activity, low expression of the DNA repair machinery, a lot of double-strand breaks, and more or less good patient prognosis. The other, okay, and if we delete in this cell population either cyclin E1 or CDK2, we get apoptosis and cell cycle arrest, and we can somehow stop the tumor progression. The other subpopulation, completely different. Yeah, we call them cyclin E1 independent hepatoma cells. They have the contradictory, they have the opposite gene signature with high activation of E-type and A-type cyclins, high overall cell cycle activity, high DNA repair activity, very low amount of double-strand breaks because of the strong DNA repair, and very poor patient prognosis in men. And if you delete, if you block here cyclin E1 or CDK2, basically nothing happens, or at best you slightly delay the tumor progression a little bit, but this will not have a therapeutic effect at all, just a delay. Okay, this is our current model, and I would like to summarize this final part. So I showed you that the liver homeostasis and the regeneration requires cyclin E1 plus one other part of the cyclin E CDK2 complex. I also showed you that the liver fibrogenesis relies on cyclin E1 and CDK2, especially in hepatic stellate cells. I tried to convince you that targeting cyclin E1 by siRNA is a suitable approach to treat liver fibrosis, at least in mice. And I showed you that HCC initiation depends on cyclin E1 and CDK2, yeah, whereas CDK2 is fully dispensable for HCC progression. And finally, I postulate that the therapeutic inhibition of cyclin E1 could be beneficial also in cyclin E1 dependent HCC subtype. But this remains to be proven. So what comes next? A very short outlook. Of course, we are now trying to apply our siRNA strategy to HCC models. I can tell you that this is challenging. This is one example where we treated the livers with DN-induced tumors with our siRNA. And the red dots are tumor nodules which have a lot of siRNA. and the blue ones, which are unfortunately the big ones, take up siRNA very poorly. So we are currently in contact with world specialists in siRNA delivery, and hopefully we can improve our delivery protocol in the near future. But what is motivating that a few months ago, the first siRNA-mediated therapy has been approved by the FDA. In other words, siRNA is no longer something very strange. It will come also into the pharmaceutical market. And therefore, maybe it's really worth to keep on track with that. And the other thing that we want to address a little bit further is the tumor environment and to analyze the role of the cell cycle machinery in cells that we have not been addressed so far. For instance, we have some preliminary data in mast cells, which are not on focus of most hepatologists. And we want also to look at macrophages and T cells. And with this, I would like to thank you for your attention. I would like to acknowledge all the people that contributed to these studies. From my lab, this was especially Roland Sonntag, but also Wei Hu, Jörg Martin Bang, and Nives Giebeler. And now the next generation will keep on track. Of course, Julia did a lot of work when she started in Aachen and Hanover with the cyclin E mice. I'm happy that we still cooperate with you and with Javier. We have a very nice cooperation with Mariano Barbaciti in Madrid. We have cooperations with the Charité and with the Dana-Farber Cancer Institute. Thank you very much. Happy to take questions. Thank you very much for your question. The answer is yes. Regarding the cyclin E1 knockout mice, we measured that the size of the hepatocyte is increasing. And we also measured, I don't remember. We also measured that the level of polyploidy is changed after knockout of either cyclin E1 or cyclin E2. As far as I remember, the cyclin E2 knockout mice, they have incredible strong polyploidy. We measured something like 16N hepatocytes, and the level of polyploidy in cyclin E1 knockout hepatocytes was very poor. So this definitely has an effect on the hepatocyte size and also on the ploidy level. I agree with you. It's a good question, and we are doing, or we want to do both approaches. We want to treat liver cancers with cyclin E1 dependent signature, with cyclin E1 only, with cyclin E1 as siRNA only. But we also want to treat some cancers with a mixture of cyclin E1 and cyclin E2 as siRNA in order to delete both components. And this would, of course, also reduce the level of cyclin E2 and hopefully target the second subpopulation of more aggressive hepatoma cells. But you are absolutely right. We need to address both components of the complex. This had something to do with, this was a very slight effect on fibrosis progression, It was a slight but significant effect. I cannot tell you what is the underlying mechanism. To be honest, we really do not understand why a knockout of cyclin E1 in hepatocytes affects fibrogenesis at all. This was unexpected. In this subset of experiments, we did not quantify the size of hepatocytes further. We did in the fibrosis paper, we looked at livers, and I can tell you that the size of hepatocytes was again larger than in the control group. This for sure. So some of the liver mass restoration was performed by hepatocyte hypertrophy. Endothelial cells? No, we did not look at endothelial cells in detail. I thank you very much for this question, and we had a lot of discussions on this topic. together with colleagues and also with reviewers. And this was the reason why I showed you this very old regeneration data in so much detail, because the effect of cyclin E1 depletion or inactivation on hepatocytes is really moderate and very slight. And I showed you in the hepatectomy model that we had at best a delay of the regeneration. And this was also true in the CCL4 model. Of course, we had in the first 24 hours or so, we had a delay in hepatocyte regeneration. They were proliferating slower, but all the mice survived. And at the end, we had the same regeneration and delayed proliferation of hepatocytes than in the control. So I believe that the modulation of the cyclins in hepatocytes will not affect the regeneration capacity in long term. It will somehow delay the process, and the hepatocytes are able to compensate partially by hypertrophy. But at the end, none of the mice ever died because of a poor regeneration. So I believe that this approach is really applicable. And of course, we have to test this also with human hepatocytes. This is something that is still open. We have to be honest here. But from the mouse data, I would predict that blocking cyclin E1 in hepatocytes should not affect the regeneration capacity too much. So it is, in my opinion, it will not be harmful at all. We do have also some concrete data on HSCs. I did not show this data. So we isolated hepatic stellate cells from cyclin E1 knockout mice and did the proliferation kinetics in vitro. And this was also accompanied by FACS analysis. And we can nicely show that these hepatic stellate cells undergo apoptosis after approximately 4 to 5 days. They will not even try to start alpha-smooth muscle actin expression because they undergo apoptosis before. And we believe that this is a very specific function of cyclin E1 in hepatic stellate cells, which is now we think that this is CDK2 dependent. And one reason could be that cyclin E2, which is sometimes a complementary E-type cyclin, is not expressed in HSC for whatever reason. So we tried a lot of inhibitor components. So there's, as I said, there's no component that is really targeting cyclin E1. There are a lot of components which state that they are targeting CDK2, and we have data that this is not true because they are all unspecific. I think for the therapeutic approach, we really need a specific targeting. Therefore, for the moment, I think this siRNA strategy could work. If we manage it to make the delivery more efficient, and we are now working with next generation nanoparticles, which will be developed by a true specialist, maybe this will work. Another thing that could work is that we develop specific cyclin E inhibitors that do not just bind to a highly conserved interaction motif, but really prevents binding of cyclin E1 to any other protein. But this is science fiction. We have to develop these molecules first. And as long as we do not have something more specific, I think siRNA will be the best approach to continue. For HCC, I also do not believe that we need a cell type specific therapy. I think a systemic therapy would work. We had a look at some other cells. We also measured the impact of the siRNA delivery on the circulating blood, and we could not find negative side effects in the gut. There's no. We have the imaging data. We did not find siRNA in the gut. Unfortunately, maybe it's also useful for colon cancer, but it will not end up in the gut, not with our delivery systems. I thank you for this question. I think that the best prognosis would be in the second cohort of patients when because of your experience, can more or less predict that this patient will most likely develop a strong fibrosis, then I think this kind of therapy would be most promising. Yeah, for patients that has already stage 4 fibrosis or so, I'm not sure if this kind of therapy will really work. Yeah, because what we showed in this intervention model is, I did not hint at this too much, that the de novo activation of other stellate cells was already blocked to almost baseline levels. Yeah, so de novo activation is blocked, but the remaining fibrosis will not revert. Yeah, so in the patient with advanced fibrosis, you need at least a second line of treatment, yeah, to get rid of the fibers that are already there. Yeah, it's good. This approach is good for inhibiting the progression, but it's not good for reversal. Unfortunately, we planned a lot of experiments with NASH models and also with alcoholic models. Unfortunately, before you came up with this nice BASH model that we saw today, the alcoholic models did not give sufficient fibrosis for us to apply the siRNA strategy. Yeah, but now with the new models that really give us inflammation and fibrosis, of course, we need to test this if this will work. And I can only speculate. Yeah, it could happen. I hope that it will not come out, but it could happen that in this ALD or NASH models, this interference with cyclin E1 will not as efficient as in the CCL. Yeah, this I don't know. Yeah, but therefore I really hope that we continue collaborating with Madrid and do this experiment. Yeah, the same is true for NASH. Yeah, as soon as we have a NASH model that gives us convincing fibrosis in the liver, we should try the siRNA treatment as well.
Then thank you very much. Thank you, Christian. Thank you, Barcelona. Thank you.